session on viral respiratory tract infections, and we'll be discussing coronaviruses in this session. So coronaviruses, these are enveloped, so indicating of the fact that they would be sensitive to lipid solvents. They have club-shaped or petal-shaped projections on their surfaces. So why are they called as coronaviruses? Is because those peplomers give the appearance of a solar corona. You can appreciate that corona type image on corona type appearance on in the image shown. So those club-shaped or petal-shaped projections or peplomers give it a corona-like appearance. They have a positive single-stranded RNA genome, and amongst all RNA viruses, they have the largest genome, 27 to 32 kilo base pairs in size. 100 to 160 nanometers the size of the virus. They have a helical symmetry and they can infect several animals. Amongst the 6 genera, coronaviruses contain alpha, beta, gamma, delta, Baffini and toroviruses. Human coronaviruses belong to alpha and beta coronavirus genus. 229E and NL63 belongs to alpha, and SARS, MERS and OC43 and HKU1 belong to the beta coronaviruses. Coronaviruses, their genome encodes 4 structural proteins: the nucleocapsid protein, which encloses the nucleic acid, the membrane glycoprotein, which is embedded in the lipid bilayer, the spike glycoprotein or the S1, which makes the petal-shaped peplomers, and a small envelope protein. So here is a view of coronaviruses. The red protein envelops the nucleic acid. This is called as the nucleocapsid protein. The spike, that is making the petal-shaped peplomers, and the glycoprotein passing through the envelope. Cultivation of coronaviruses is very difficult. In fact, they grow only on organ culture that is derived from tracheal rings. So they grow on human tracheal ring agar cultures. And exceptions to this cultivation is SARS and MERS. They can be easily grown on viro cell lines. Human coronaviruses cause common colds. 10 to 35% of common colds are due to human coronaviruses, depending upon the season. And increased prevalence of human coronaviruses is seen in late fall and winter and early spring. They are transmitted via aerosols, infect the ciliated epithelial cells of the nasopharynx, damage these cells and lead to symptoms of common cold due to the inflammatory response that develops against them. Incubation period of coronaviruses is 3 days and they have a shorter course, 6 to 7 days. Some studies have shown that they are associated with a nasal discharge more than rhinoviruses. Sometimes they can cause pneumonias and exacerbations of COPD in adults. Lab diagnosis of human coronaviruses generally is not required. RT-PCR can be used, ELISA for serology, or indirect fluorescent antibody testing for serology culture is difficult. Coming to special coronaviruses, SARS and MERS. SARS coronavirus was first described in an outbreak in southern China in 2002 to 2003. This SARS virus spread to cause more than 9,000 cases spread over several countries in the world, in Asia, North America and South America and Europe. But more than 90% of these cases were reported from China and Hong Kong. There were at least almost 800 mortalities in this outbreak in 2002 to 2003. Reservoir for the SARS coronavirus is fruit bats, and they are likely spread from palm civets, contacts with humans in animal markets. There is also evidence of human-to-human transmission. This outbreak ceased in July 2003, and in 2004, just 17 more cases were reported, and since then, for the last several years, there has no case of SARS been reported from the world. It is transmitted via airborne droplets, and one outbreak in Hong Kong from an apartment complex suggested that fecal-oral transmission could also be one of the routes, but it is still being investigated. SARS infects the respiratory tract epithelium, leading to viremia, viruria. It is also present in the patient's stool. The virus persists in the respiratory tract for 2 to 3 weeks. Incubation period of SARS is 2 to 7 days. Malaise, fever, headache, myalgias initially, followed by a non-productive cough, dyspnea. These appear 1 to 2 days later. 25% of inpatients present also with diarrhea. X-ray chest shows patchy infiltrates, which may be peripheral, lower, or concentrated in the lower lung fields, or they may be interstitial infiltrates initially and progressive to involve the whole lung fields, leading to diffuse infiltration. Some individuals who have certain risk factors develop more severe disease, so they are at risk of developing acute respiratory distress syndrome or with multi-organ failure. These risk factors are age over 50, diabetics, COPD patients, serious disease, hepatitis, and pregnancy. Such individuals would obviously develop multi-organ failure and lead to death. So case fatality rates of SARS is about 9%, and it has been observed that children develop less severe disease. Lab diagnosis of SARS, lymphopenia is seen in about 50%, thrombocytopenia, raised LDH, creatinine kinase, SGOT, and SGPT, and RT-PCR of the respiratory tract specimens as well as of plasma is done very sensitive. Early in the disease, we will detect them in plasma and respiratory tract. Can also detect this, do RT-PCR in urine and stool later. We can culture it on Vero cell lines. ELISA and indirect fluorescent antibody testing can be used to detect antibodies which appear after 1 month. No specific therapy is known. Supportive care is done. Ribavirin has least effectivity. Vaccine is under development. Another important member of coronaviruses is MERS, Middle East Respiratory Syndrome coronavirus. Since September 2012 to September 2017, WHO has reported 2,121 cases of MERS coronavirus with at least 740 deaths, and it has been reported from 27 countries of the world, including in America, Malaysia, Indonesia, Korea, etc. But it has not yet been reported from India. There is always a history of contact or travel to 6 countries of the world: Jordan, Oman, Qatar, Kuwait, UAE, or Saudi Arabia in patients of MERS, and approximately 82% of MERS have been reported from Saudi Arabia. But the largest outbreak that was outside Middle East occurred in the Republic of Korea, which resulted in 186 cases with 38 deaths. Reservoir for MERS is bats, probably spread to humans via camels. The route is exactly not clear, either by ingestion of raw milk, camel milk, or by ingestion of camel urine or undercooked camel meat. Human-to-human transmission has been shown positive. It requires close contact, such as caring for or living with infected individuals, and contact with respiratory secretions. Incubation period of MERS is 5 days. Mild respiratory symptoms or asymptomatic cases may be seen, but patients who are symptomatic develop cough, fever, dyspnea associated with diarrhea and vomiting. ARDS and respiratory failure develop in 1 week. Risk factors are absolutely same as for SARS coronavirus. Renal failure is often seen because MERS coronavirus receptors are also located in kidneys. Pericarditis has also been reported in some patients. Fatality rates range from 27 to 35%. And diagnosis: lymphopenia, neutropenia, thrombocytopenia, raised LDH. Apart from that, specific diagnosis can be done by taking serum or respiratory secretions or stool specimens. We can do PCR, we can culture it on monkey kidney cell lines or Vero cell lines, and serology can be used to detect antibodies in the serum. No established antiviral therapy is there yet, and vaccine is under development. So here we have a question. A paid traveler returning from Mecca presents with pneumonia, fever, and cough. What is the best test to diagnose MERS coronavirus? So best test would be MERS coronavirus PCR. Following are common characteristics of coronaviruses, except they contain the largest genome of RNA viruses. They are distributed worldwide, have cross-reactive antigens with influenza, have a helical symmetry. Out of these, the only one which is wrong is C. They do not have cross-reactive antigens with influenza. They belong to corona, whereas influenza belongs to orthomyxoviruses. False about MERS. It is a coronavirus. Correct. The symptoms of MERS appear within a day or two. That is not so. It's a longer incubation period. Fever is the most common symptom observed in MERS. Yes, that is true. That's the most common symptom. MERS usually affects individuals who have a pre-existing illness like diabetics and heart disease. That is also true. So the only one which is correct is the wrong incubation period. It is not 1 to 2 days.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future perhaps as a therapy. This animation will introduce you to the principles of RNAi involving 2 important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs, or double-stranded RNA, is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaut protein. One strand of the RNA is selected and remains bound to Argonaut. This is called the guide strand. The combination of the RNA and Argonaut, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaut catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonauts and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.
Thank you very much for giving me the chance to talk about our messenger RNA-based vaccine studies against SARS-CoV-2. A number of years ago, Drew Weissman and I developed a messenger RNA-based vaccine platform that we've been using for the vaccine development against infectious diseases, and we recently started some cancer vaccine-related studies as well. So let me show you a cartoon about the platform and just tell you very briefly about mRNA production. So the messenger RNA is in vitro transcribed from a plasmid DNA template that has the antigen coding sequence. It also encodes the 5' and 3' UTR elements of the messenger RNA, and these are absolutely critical for mRNA's increased stability and translatability. The plasmid also encodes a 101 nucleotide long optimized poly-A tail that further stabilizes the RNA. So once we perform the in vitro transcription reaction with the 4 building blocks of the RNA and the RNA polymerase and the linearized plasmid DNA template, we add the 5' cap bond structure to the RNA that further stabilizes it, and it's also critical for high level of translation. It's been known for a long time that RNA polymerase makes mistakes during messenger RNA synthesis, and the byproducts are primarily short double-stranded RNAs. And these short double-stranded RNAs, as well as uridine, are recognized as pathogen-associated molecular patterns by the immune system, and they can induce unwanted immune responses, such as very strong inflammatory responses, type 1 interferon production, and eventually the inhibition of translation. So from the messenger protein production from the messenger RNA. So obviously this is something that we don't want. So we purify the in vitro transcribed messenger RNA to get rid of this double-stranded RNA contamination, and we also replace uridines with 1-methyl pseudouridine that is further contributes to the high translatability of the messenger RNA. Once we make the RNA that is purified and nucleoside modified, it gets formulated into lipid nanoparticles, and these special lipid nanoparticles protect RNA from extracellular degradation, and it also helps to induce potent immune responses. So we call this vaccine platform nucleoside modified mRNA LNP vaccine, and I just listed some of the advantages of using this vaccine platform. Besides potency and safety, I'd like to mention that it's very important that the production of these mRNA vaccines is very simple, similar to DNA vaccines. It's completely these vaccines are synthetic. The production is sequence independent, so it's possible to use the same infrastructure and the same strategy to produce any RNA vaccines. And there are at least 3 big RNA companies that can produce millions of those vaccines if necessary. So we use this vaccine platform to design and produce 4 different RNA vaccines. In the first round, just 4 different vaccines against SARS-CoV-2. We would like to target the spike glycoprotein of this coronavirus, and we designed 4 constructs: a full-length S protein encoding mRNA. The second construct, it's also a full-length, but with a mutated furin cleavage site. We have a third construct that has 2 mutations in the S2 region. Valine and lysine were mutated to prolines, and we expect that in these 2 constructs, the furin mutant and the 2P construct, we can keep the spike protein pre-fusion state, and we can hopefully induce better immune responses compared to the wild-type construct. And we generated a 4th construct as well. This is a short one, encodes only the receptor binding domain of S protein. So the second one and the 4th one are already in mice, and I can share you some very, very limited immunogenicity data. So the experimental setup was very simple. 8-week-old biopsy mice were injected intramuscularly or intradermally with a single dose of 3 or 30 microgram of mRNA LNP vaccine, and the control group received Firefly luciferase encoding RNA, and we keep bleeding-based mice, but I can share you some ELISA data at a very early time point, 2 weeks post-immunization. So after a single intramuscular immunization with 30 microgram mRNA LNPs, we could induce pretty robust immune responses as measured by S protein specific ELISA. So the ELISA titers are in the hundreds of thousands. That is actually pretty good for 14 days post-immunization, and the lower dose 3 microgram also induced pretty decent ELISA titers. We obtained similar data after intradermal immunization with 30 microgram. So the take home from these experiments is that the vaccine is certainly, or actually both SARS-CoV-2 vaccines are immunogenic in mice after intramuscular and intradermal immunization, but it will be much more important to look at the neutralization titers. So Paul Bates is performing pseudovirus neutralization assays, and this will be absolutely critical. We would like to follow antibody titers in these injected mice to look at the durability of antibody responses. In previous studies with influenza and Zika vaccines, we demonstrated that this vaccine platform induced durable antibody responses many times after a single immunization. We would like to expand these studies, and I've been working with Michela Locci on a bunch of different projects, and we would like to team up and look at the T follicular helper cell, germinal center B cell, memory B cell, and plasma cell responses in the context of the new coronavirus. I'm looking forward to it. It would be very important to demonstrate the protective efficacy of these vaccines. It won't be that easy because the best animal model would be a human ACE2 knock-in mouse. But these mice are not available now. So we will probably go with a viral challenge in wild-type animals, probably in wild-type BARB-C mice. This is what we did in our Zika studies, and actually we could detect differences between the Zika vaccine and the control vaccine immunized animals. So we hope to get similar data in these experiments as well. And in a week or two, we will start some large animal studies with Bart Haynes from Duke University. We've been working with Bart for a long time, primarily on the development of HIV vaccines. So basically, this is what I could show you, and this is just a small team of people who generated the data I showed you. And again, we plan to expand, and again, Paul Bates is included in the new studies, Michela in some more basic science studies, and I'm happy to expand and start collaborations with multiple people. Thank you very much.
American company Moderna has released first batch of vaccine against novel coronavirus, against this SARS-2 coronavirus. This vaccine is actually in phase 1 clinical testing. What I mean by this is that before our drug is launched, firstly it is discovery and preclinical phase. In this phase, it is tested in labs and on experimental animals for observe any adverse effects in animals, right? And after that, human trials or clinical trials starting from phase 1. This drug or vaccine, it is given to very small number of volunteers, 20 to 100 volunteers, and they are observed to see if any adverse effect develop. Then gradually bigger population is chosen, 100 to 300 volunteers in phase 2 and 300 to 3000 volunteers in phase 3. And if all goes well, it can be finally approved by FDA for public use. Then it is marketed for public use and it is launched. So right now this vaccine, it is in phase 1. So it may take up to year or so before it would be available for general public, right? The interesting thing about this vaccine, which is launched by Moderna, is that it is mRNA vaccine. Unlike the traditional vaccines that are either live attenuated virus or protein subunit vaccine, it is mRNA vaccine, And mRNA or messenger RNA, as you know, it codes for unfused, stable unfused S protein or spike protein of coronavirus. And by the way, this vaccine is called mRNA 1273, right? So what happens is that, as I have told you in the lecture, visual lecture on replicative cycle of coronavirus, that here is the coronavirus and here is its S protein. What will happen that through its S protein, it will bind with ACE2 receptor of alveolar type 2 alveolar pneumocyte, and this will lead to fusion of this viral envelope with cell membrane, and it will enter into cell and it will infect the cell. So with this vaccine we target this step, right? So we target this step with this vaccine. So today we will discuss that how this mRNA vaccine will work. Hello, I am Dr. Azaz from medicovisual.com. Welcome to this visual lecture. So this vaccine, let's suppose this vaccine is given through intramuscular injection in deltoid. So I am gonna enlarge this part and we will see that what is going on inside this deltoid. So here is the vaccine and it has been dropped inside the interstitial space, the space between cells, right? So this vaccine, it actually contains mRNA which is encapsulated by lipid nanoparticle. So mRNA is present inside a lipid nanoparticle. Why do we need this lipid nanoparticle? Because if we insert a naked mRNA, if we inject a naked mRNA, what will happen that there are some hostile RNAs is present in the extracellular environment which will digest this. RNases mean RNA digesting enzyme, they will digest this. Secondly, mRNA, we need mRNA inside the cell to function, right? There is no use of mRNA outside the cell. It may even trigger immune response against itself, which will render the vaccine useless. So mRNA by itself cannot enter into cell. So this lipid nanoparticle, it will be used as a delivery vehicle. It will be used as a delivery vehicle to safely deliver this mRNA inside the cell. It will release the mRNA inside the cell and it will be digested here. So now we got the mRNA. This mRNA vaccine, it actually imitates an actual virus infection. Just like an actual coronavirus which infects the cell and injects its mRNA inside the cell, it is acting just like that. It is so natural, right? But the difference is that it codes only for S protein. It is not coding for any other pathogenic proteins. So of course it cannot cause the disease, but it can trigger immune response, right? So here is the mRNA. It will use cellular protein synthesizing machinery, so to form what? To form S protein or spike protein of coronavirus. This spike protein will be processed by intracellular proteases, and this, after processing, the fragments of S protein, they will be presented on MHC1 protein. They will be presented on a special receptors called MHC1. Actually all nucleated cells have to present or express their cellular proteins on MHC1 receptor. This is to show the immune system that what is going on, what protein is being synthesized inside these cellular factories. Of course, normal cell will present normal cellular proteins on MHC1, and viral infected cell, or in this case vaccine infected cell, will of course present MHC. On MHC1 it will present viral proteins or fragments of S protein in this case. Some of these proteins will be presented on MHC1, while most of it will be packed by Golgi apparatus, processed by endoplasmic reticulum, and will be packed by Golgi apparatus, and these S proteins, they will be secreted outside the cell, right? So they will be secreted outside the cell. Now let me tell you something interesting. Billions of cells, billions of cytotoxic cells with millions of different specificities are wandering in your body. They are wandering in plasma and they are trying to find their target. Somehow a cytotoxic or killer cell, cytotoxic T cell, which may be specific for this S protein, it may come across this S protein randomly, and it will bind with its T cell receptor, with its TCR or T cell receptor. It will bind with this MHC1 bound S protein fragment, MHC1 bound antigen, and it will become active. This cytotoxic T cell, it is also called CD8 positive lymphocyte, because it is a lymphocyte which expresses CD8 protein, right? So it is cytotoxic T cell. Cyto means cell, toxic means killer. So it is cell killer. We will see what it does. So this cytotoxic T cell will recognize it and will become active. It will become happily active. Not only cytotoxic T cells are present here in our body, we also have B cells. So B cells or B lymphocytes, they are not fussy about MHC1. So they can directly recognize the antigen and become active, right? So cytotoxic T cell, which are responsible for cell mediated immune response, and B cell, which are responsible for humoral immune response or antibody mediated immune response. So once they will bind with this antigen, B cell, they will bind with this antigen, they may become activated and they may convert into plasma cell, and they will secrete antibodies specific for this S protein, right? Now we also have some macrophages wandering here and there. So these macrophages, they will recognize these foreign proteins. What are these foreign proteins? These S proteins. They will recognize these S proteins and they will engulf these S proteins, and then they will present these S proteins on MHC type 2 receptors, not on MHC1, on MHC2 receptor. MHC1 receptor, here is the MHC1 receptor, and they are they are expressed on all nucleated cell, while MHC2, they are expressed only on professional antigen presenting cell, for example dendritic cell and macrophages. MHC1, they are involved in expressing endogenous proteins, while MHC2, they are involved in expressing exogenous proteins. What are endogenous proteins? Endogenous proteins are those proteins which are synthesized and processed inside the cell. They are synthesized inside the cell, while exogenous proteins are those which are taken up by macrophages from the extracellular environment. From the extracellular environment, they are engulfed and presented on MHC type 2. So as soon as they will express these antigenic fragments on MHC type 2, S protein fragments on MHC type 2, they will take them to helper T lymphocyte, and here come the master of immune system. Helper T lymphocyte is master regulator of immune system, or we also call it that it is orchestrator of immune system. It is most important cell in immune system. So helper T lymphocyte is also called CD4 positive T lymphocyte because it expresses CD4 receptor. So it's with its T cell receptor, it will recognize and bind with MHC2 presented antigen, MHC2 bound antigen. Now what it will do? Now it will give some orders. to cytotoxic T cell as well as B cell. It will release certain cellular messenger or cellular growth factors, or we call them cytokines. It will release cytokines which will cause growth and proliferation of cytotoxic T cell as well as growth and proliferation of B cells. It will cause what? It will cause clonal proliferation. And what I mean by clonal proliferation is that it will make lots and lots of copies of this specific cytotoxic T cell. It will make lots of copies of cytotoxic T cell that is specific for a particular antigen. It will make copy of cytotoxic T cell which is specific for this antigen, this S protein antigen. And it will also cause clonal proliferation of B cell. And it will also release cytokines which act on itself, and it will make copy of itself as well. So when it will make copy of itself, it will lead to massive immune response, and it will lead to amplification of whole this immune process. Now also it will activate these cells, cytotoxic T cells and B cells. So what will happen? That cytotoxic T cell will release granzymes and perforins that will destroy these vaccine infected cells, right? And after activation, B cells will be converted, some of these B cells will be converted into plasma cells, and plasma cells are antibody generating factories. So plasma cell will do what? It will generate lots of antibodies which are specific for S proteins. Now these are the effector cells, the cytotoxic T cell and this cell, plasma cell, these are effector cells. They are doing something in response to this. But some of these cells out of these clones, they will remain dormant, and they will act when a subsequent infection occur, when subsequent exposure to antigen occur, when an actual virus infection occur, then these dormant cells will become active and they will do something. They will produce immune response. These dormant cells are called memory cells. So we have memory T cells, we have memory cytotoxic T cells, we have memory B cells, and we will have lots of S protein specific antibodies, and we also will have memory helper T cells. So we have got all these things, right? So what will happen when an actual virus enters? So when actual virus tries to infect, these antibodies will come to our rescue, and they will prevent fusion of this S protein with ACE2 receptor of type 2 alveolar pneumocyte. It will prevent fusion, right? Also these antibodies, they may trigger complement system and some other pathway. I won't go into detail, and they may directly cause killing of this virus particle, right? And even if somehow this virus, it may escape this antibody mediated immune response, and it enters the cell, and it starts generating and expressing its viral proteins, and it starts making lots of copies. But because we have memory cytotoxic T cell, they will quickly and swiftly react. And before this virus can release these, assemble and release these virus particles outside the cell, cytotoxic T cell will quickly come and destroy this cell and everything inside it. So of course virus will not be able to infect. So in this way, due to humoral response or antibody response as well as cell mediated response, this vaccine can prevent the actual virus infection, actual coronavirus infection. Now here I would love to highlight another point, that how this mRNA vaccine is better than traditional protein subunit vaccine. Let's suppose if we give an S protein directly, we are not giving mRNA, we are giving S protein of coronavirus directly. It may be recognized by B cell and it may be engulfed and recognized by macrophages, and antibodies will be produced. But because it is not mRNA, can it be presented on MHC type 1? Of course not, because MHC type 1 is only concerned with presentation of endogenous protein, and this is not endogenous protein, this is exogenous protein. So will there be a significant cell mediated immune response or cytotoxic immune response? Of course not. So this immune response, cell mediated immune response, will be negligible. But of course we will have humoral or antibody mediated immune response, as well as of course we will have helper T lymphocyte, right? So because we are not getting a strong cell mediated immune response, it is not an ideal vaccine, this protein subunit vaccine, because cell mediated immune response is very important in clearing viral infections, in clearing virus from our body, right? Now what about mRNA vaccine versus live attenuated virus vaccine? Live attenuated virus is basically a living virus, a live virus, but it is weakened. It is so much so, weakened so much so that it is no more pathogenic. It can trigger immune response, but it cannot cause the disease. We use certain chemicals to weaken this virus. Now the problem with this is that in certain rare circumstances, especially in immunocompromised people, it can suddenly turn into pathogenic strain and it can cause disease. So that is a real side effect, especially it has been observed with polio vaccine, right? So this was about this vaccine. Now this vaccine, it seems quite promising, but as of now its safety profile is not clearly elucidated, right? So thank you for watching this lecture. If you like this visual lecture, please like this video, please subscribe to my channel, and please give your precious comments regarding this lecture. Thank you so much for watching this lecture. So
thank you very much, PGA, and good morning, good afternoon, and good evening, wherever you are in the world. And thank you for joining us for this special session of the United Nations General Assembly in response to the COVID-19 pandemic. My name is Melissa Fleming, and I'm the Undersecretary General for Global Communications here at the United Nations. And I have the honor to moderate this high-level panel. And this panel is going to focus on the UN system's response to the COVID-19 pandemic. And in today's discussion, we are going to outline what worked well so far in the UN's response, Also, where there were operational policy gaps, and very importantly, were there lessons learned. It will highlight actions needed as part of the ongoing humanitarian, health, and human rights responses, as well as the launch of economic and social measures to mitigate the terrible suffering that we've seen and to help the world, especially the most vulnerable, recover from the pandemic. The PGA has already introduced our distinguished panel, and so I will go right to the session and the questions. And my first question goes to Dr. Mike Ryan, who is in Geneva at WHO headquarters. Mike Ryan is the Executive Director of WHO's Health Emergency Program. Mike, it has been almost 1 year since the COVID-19 pandemic started. And as the virus continues its deadly onslaught, surging for a second wave in many countries, could you describe what went wrong? And what are the priorities for WHO in getting through this dark winter? Is the vaccine our only hope? And finally, how is WHO working with other UN agencies to help the people who are most at risk?
Hi, Melissa. Good morning, good afternoon, good evening, everyone. Hello to my fellow panelists. I can see David, Mark, and Michelle, and others online. And let me just first recognize the pleasure and honor it's been to work with UN agencies, NGOs, and others across the board over the last 10 months. It's been a long journey, and we have a long way to go. But it's been wonderful to find ways of working together even more collaboratively and more effectively as we've all faced the horrors of this virus, not only in terms of health, but in terms of social, economic, education, human rights, travel, and other impacts that it's had around the world. With regard to where we are right now, and with the prospects for vaccines, we are still in a very, very dangerous and difficult situation. We're adding 4 million cases a week, and we're adding large numbers of deaths, up to 70,000 deaths a week. Some countries are still in the throes of major transmission, particularly in North America and Europe. Other parts of the world have stable transmission, but there are still countries within those regions that have significant and difficult issues with either continued transmission or returning transmission. And this epidemiologic situation has not stabilized. And let me be clear, this has not stabilized. And countries that are out of major transmission may jump back into it, depending on what they do in terms of prevention, control, and mitigation. And those that are currently in intense transmission are going to find it very difficult to exit without consistent, comprehensive strategies that are properly directed to controlling the spread of the virus and its impact on the health system and the economic system. With regard, and I'm sure our colleagues in the next session will speak to the advent of vaccines. We have hope. We will be able to add vaccines to the current control measures. But let me be clear, vaccines does not equal, or vaccination does not equal zero COVID. We must add vaccines to the existing arsenal of measures that we have in place. If we add vaccines to our current toolkit, we have a significant opportunity to suppress transmission, control transmission, and potentially stop transmission. But if we can stop the deaths, if we can take the pressure off our health system, if we can control the transmission dynamics, then our economies and our peoples and our society can return to what we would recognize as normal, or a new normal at least. That is going to require a huge effort, and we thank our colleagues in other UN agencies and all around the world in ACT, and particularly our colleagues in UNICEF, who are leading the drive to get countries ready for vaccination. Having vaccines is one thing, as you know, Melissa. Vaccinating people is an altogether different challenge. And therefore, the hope of vaccines is wonderful, and we should not lose that hope. But we must sustain the current effort. We must continue to contain and mitigate the impact of this virus. We must plan and be ready to deliver vaccines in a fair and equitable fashion and so many others of our UN colleagues are so important. And you and I have spoken about the need for driving vaccine demand and reducing vaccine hesitancy. That's a challenge in community engagement, in communications, and in social marketing, for which other UN agencies are much better placed and skilled than WHO. And I think this response has shown that the UN has the DNA for complex responses in multiple countries. But we have to share that DNA. We have to bring together the pieces, the chromosomes that make us a whole, that give us the power to act collectively in the service of our member states. And certainly so many of the initiatives over the last number of months, obviously the leadership of the Secretary General and the DSG, of the various heads of the agencies, but importantly the work that we've done together in the UN Crisis Management Team, in the IASC, and on the COVID-19 mechanism there that was co-chaired by Mark and Sosé Fall, who's here with me today, the socioeconomic response coordination, the humanitarian response, and so many different mechanisms and instruments were utilised. And above all, I think the work that Robert Piper and others have done in bringing together the UN country teams, the leading role of the humanitarian and resident coordinators, joining the efforts of the whole UN at country level and presenting that as a joint effort to our member states, developing the country partners portal, developing the UN supply chain mechanism, delivering on a joint service for communications and RCCE through the Red Cross and the work of UNICEF and ourselves and yourself. So many initiatives that have given us the opportunity to work better together, to leverage off all of the things our comparative advantages in each area. I have really been humbled by the way in which people have put aside any competition, any rivalries, any differences, and acted in the true interest of our member states and their citizens. I am proud to be UN. We are proud to be UN, and I'm sure others will speak to the specific issues at hand. But we have all worked, and beyond the UN system, our NGO colleagues at country level, INGOs and local NGOs, our scientific collaborations all around the world, the private sector who've joined us in many of the efforts we've done in science and communications. This has been an all-of-world effort, one of the finest in my view in history. And despite the problems and the issues and the challenges, Melissa, and yes, there are, we were not prepared. We were not ready to deal with this crisis. We had not invested enough collectively in preparing for a crisis like this. We hope we have all learned that lesson, and we will now follow through with the necessary investments. But rather than end on a negative point, I would say that while we were not ready, the world's response collectively has shown what can be achieved when we work together, but also has really, really highlighted when we have disunity, when we have lack of cohesion, when we have lack of clarity, lack of consistency. Doesn't matter if you're at local, national or international level. Without that clarity and unity, we do not do well. And therefore, the word united in United Nations means more than ever before. Back to you.
And there are today people who are still actually anti-science. A whole movement called the anti-vaxxers, who refuse to acknowledge the evidence that vaccinations have eradicated smallpox, and who, by their prejudices, are actually endangering the very children they want to protect. And I totally reject this anti-scientific pessimism. I'm profoundly optimistic about the ability of new technology to serve as a liberator and to remake the world wondrously and benignly. Indeed, in countless respects, technology is already doing just that. Nanotechnology, I mentioned earlier. Revolutionizing medicine by designing robots a fraction of the size of a red blood cell, capable of swimming through our bodies, dispensing medicine and attacking malignant cells like some Star Wars armada. Neural interface technology is producing a new generation of cochlear implants. I think of new tools that we acquired, but over which we, the human race, had the advantage, which we controlled. And that is not necessarily the case in the digital age. You may keep your secrets from your friends, from your parents, your children, your doctor, even your personal trainer. But it takes real effort to conceal your thoughts from Google, from Google, from Google. And if that is true today, in future, there may be nowhere to hide. In future, there may be nowhere to hide, nowhere to hide, nowhere to hide, nowhere to hide. Smart cities will pullulate with sensors, all joined together by the Internet of Things. Bollards communing invisibly with lampposts. But this technology could also be used to keep every citizen under round-the-clock surveillance. A future Alexa will pretend to take orders, but this Alexa will be watching you, clucking her tongue and stamping her foot.
out, we're told, as of Monday in this country. Let's get the thoughts of Dr. Mike Yeadon. He's a former Chief Scientific Advisor for Pfizer, and we've spoken to him many times over the show, many times on Talk Radio, about concerns about testing and about, what I suppose what a lot of people say, what's really going on behind the scenes when it comes to the scientific and medical management of the coronavirus virus. Good morning to you, Mike.
Good morning, Julia.
Good to talk to you. Now, you've raised a lot of concerns about testing, which is very much your area of expertise from your many, many years working at companies like Pfizer and setting up your own company involving with testing and lab work and the like. What are your thoughts on the rollout of this vaccine? Because there are an awful lot of people who've got concerns about the testing and concerns about government policy who are raising what a lot of people would regard as conspiracy theories about how, this vaccine, it's all about making money, it's not going to do anything good, it's about controlling us or the like. Do you sign up to any of those conspiracy theories?
No, I don't. So, Julia, first, well, thank you for having me again. First, I was the Chief Scientific Advisor for Respiratory at Pfizer. I would love to be. Anyway, so first I will say, because I'm going to say some things that aren't popular, I'm going to make it clear I'm not an anti-vaxxer. I've spent my entire working life in the pharmaceutical industry, and including to today, where I advise biotech clients. I've had all the vaccines myself, so has my wife, so did our children when they were small, and as evidence that I stay current and believe in well-developed vaccines that meet a medical need, when my children who are now in their 20s, when they were in their teens and the most recent HPV vaccine arrived, I looked at the data, talked it through with my children, and advised them to make their own decision. So I'm pro-vaccine if it's developed well and meets a medical need. So where am I on this vaccine then? I would say, and this comes from experience not of vaccine development, which I don't have, but 32 years in drug development, R&D. So safety is the most important factor. You can't compromise on that. The next is how well does the new medicine work? And when you've understood both those, the safety as it were as a risk, the effectiveness, the benefit, you can look at risk-benefit, and even that ratio will change depending on the unmet medical need, the amount of benefits that you're going to get, not just effectiveness, but is the person suffering from the condition you're going to treat them? And so I would say I do have a concern because in the main, young, healthy people they're not likely to get severely ill or to die with this virus. Now, some people do. There were clearly some cases in the spring, but certainly looking in the autumn now, it does seem to be back to the elderly and already ill that are most at risk. And so I would want to see it offered to those people. And I'm very concerned about a wide rollout for this reason, that we don't yet know enough about the long-term safety. That's not controversial. We've just not been in people very long. And perhaps a bit more controversial, I don't think we know much about the effectiveness, because the trial has only studied whether or not people became PCR positive. And people will know I have concerns about that. And I think they're valid. They haven't looked at full-length viral sequencing or cultures. So we've just got PCR positive or not. I do think the vaccine has done something to that endpoint. I don't think we can be clear that that will lead to reductions in severe illness or death.
Well, I think they have claimed that that is what they're looking at. Now, again, we're told this vaccine and other vaccines 90% plus effectiveness, even in people of the older age category who are most at risk. So am I right in thinking your argument is that, look, yeah, people should, the vaccine is definitely good for people who are the older age categories, people in their 90s, their 80s, their 70s, possibly even their 60s, people who are at higher risk, those who are vulnerable, but that you don't think there should be a mass rollout vaccination campaign for everybody else who is at, and we've seen the statistics, at much, much, much lower risk of dying.
Yes, you've summarised it very well, but I would just come back to you said, you invited me to agree that it's very effective in this older group. I would say we don't know that yet. I do accept, and Pfizer has said that is their intention, but the basis of approval of the drug, we've not yet seen all the information, the basis of approval by MHRA yesterday, presumably, was on the basis of reduction in becoming PCR positive, that is, as a surrogate for infection. So they don't even have solid data that it reduces infection. I think it will do that, But will it reduce severe illness and death? I don't think anyone can say.
And it's going to be really crucial that we do see the evidence on which the MHRA, this is the Medicines and Healthcare Products Regulatory Agency, the basis on which they've made that decision, so that people, I mean, that's just good practice, isn't it? That everyone can be reassured.
It should be released. Normally, I don't know if it's true, uses this phrase, but they usually have an SBA. It's the summary basis of approval, and it can be written quickly because it's a summary. So I would want to see the vaccine equivalent of an SBA this week, please. It's really quick to do. You can tell us what was the basis. Please do that.
I mean, are you concerned at all about the talk of this idea of a COVID passport certificate? So you get your vaccine, you've got a certificate, and then that will, allow you. Qantas Airlines have already said you're not going to get on one of their planes next year without a vaccine certificate. I'm thinking, well, I won't be using Qantas then. Thank you very much. No. But you wouldn't be able to say go to a sports stadium. Wouldn't be able to go into certain restaurants. Private businesses would be allowed to say, I need to see effectively your health records before I allow you access to my business. What do you make of that? And even some people talking about how this needs to be a mandatory vaccine.
I know. Well, certainly mandatory is extremely problematic. I hope that listeners know or watchers know that because of some very unpleasant things that happened in the Second World War, all the countries of the world decided after that they would put in place a convention that said that the human body is sacrosanct and that no one shall perform a medical procedure on it without the consent of that person. Obviously, there are some exceptions, people who don't have agency or capacity. But for the rest of us, it's simply illegal under international law to which we are a signatory to go to mandate. And that's also true of coercion. So I would advise the government to persuade people to take it on the evidence. So if you provide the evidence that this is good for you, you'll take it. That's as far as we should go. And where it comes to vaccine certificates and things, I'm worried about that because I think it will become coercion. And also, why don't we just be sensible about this? It's a nasty vaccine, a nasty virus. It definitely has killed a lot of people, and it still represents a threat if you catch it and you're not immune. But it's a worse version of flu in most people who are old, and a less severe risk for those who are much younger. I don't understand why we're getting quite so hyperventilatory about this, but that's politics, not science.
Yeah,
I'm uncomfortable.
But that's it. Yeah. But the politics and science have become very intertwined, haven't they? Because with this idea, a lot of people seem to be caught up with this idea that you could go to zero COVID, you never have a single death. People saying one death is too many. Well, it's seeming not to realise that 1,700 people die of various things every year. We are seeing, are we not, in the data right now that we've basically had a complete collapse of flu deaths. Now, this may due to the fact that people are self-isolating, people are social distancing, we've been in lockdown. But a lot of people think lots of experts think that it may well actually be that people are catching the dominant virus going around, which would be coronavirus, not flu. No. But also that a lot of the deaths right now may actually be called COVID deaths, and we have to be very careful about what those deaths are registered. They are deaths that are registered involved. The technical thing is they involve COVID. So we are seeing, when we see these death numbers every day on the news, they are for deaths that have been registered where someone has died with COVID, not necessarily of COVID.
Yes. No, I can comment on that. So people who are followers of my good colleague, Dr. Claire Craig, who is a diagnostic pathologist. So she's done all the work that I'm going to mention. So yes, I don't want to put words in her mouth in case I get it wrong, but it's certainly true that there is over assignment of deaths to COVID. So not all of the people who have got a COVID death, that is, they died within 28 days of a positive PCR test, not all of those people died because of the virus or even with it. Sometimes they'll be false positives and so on. But I just want to make a quick comment. You've said I talk about testing, I do, but my training also includes immunology, and we seem to have forgotten immunology. I'm convinced, and I hope I can do it in 3 or 4 sentences, that to say the country's broadly at herd immunity. So there's been extensive papers published to show that people had prior immunity because their bodies had seen a related virus, the common cold causing coronaviruses. That's not controversial, even Public Health England has now published on that front. I believe we're at herd immunity as evidenced by the lack of COVID deaths in London right now, which were about 200 to 250 every day. 7 months ago, I checked 3 days ago, it was 9. So that means it's 95, 96% lower. Now, there are some areas that were hit hard in the spring and are still being hit hard, and it's Liverpool. But a friend of mine, Joel Smalley, has looked at this, and it's the same pattern. There are districts and regions in the northwest that contributed the deaths in spring, regions A, B, and C. I don't remember their names. But in the autumn, it's not A, B, and C. All the deaths come from D, E, and F. So I think there's strong evidence of herd immunity. I advise people to go and look at that. The reason this is important is not that we don't need a vaccine, but it means there's a weaker case still for, as it were, trying to throw ourselves in front of the train by using a vaccine, that's the only thing that'll stop it. I think it's mostly self-limiting, and definitely still people catching it and dying, and mostly the elderly and ill. I would want to see them offered the vaccine first, and I think we should just take a step back and not be so, frankly, so hasty, because it's risky.
Okay, I really appreciate you joining us, Dr. Mike Yeadon, former Chief Scientific Advisor with Pfizer. I know people are being very eagle-eared and eagle-eyed when we have guests like Mike Yeadon on, putting alternative points of view. But again, an awful lot of the things that he says, it's amazing how often, a few months down the line, the likes of Public Health England and others do accept, including the item about T-cell immunity, which he was talking about there. But again, we're saying that actually this is a vaccine that should be rolled out to those who are most vulnerable to the virus, but perhaps not necessarily hastily rolled out to those who are not most vulnerable. That would be those of us under 60 who do not have underlying health concerns. That his point of view. We put the points of view of many different experts on the show. We're not afraid of debate here at Talk Radio, as you'll know.
You know, in order to, let's say, write off your position or your thoughts, a lot of folks will call you anti-vax, as if being skeptical, which I believe obviously people have the right to be, but you have to understand what we're dealing with. Let's just break it down just for a time being. We have COVID-19, which is a coronavirus. They just found out about this, well, at least on this side of the planet. You're talking about this year. And now you're telling me that they have a vaccine that has been tested top to bottom. We know it hasn't, because when it comes to a more extended period of time, they have not done that because they can't do that. Now, there's other coronaviruses that don't have cures. We've talked about John Ioannidis and his, talking about vaccines and how they would do vaccines for that they thought were going to work, like for coronaviruses in cats, for example, and the cats were worse with the vaccine. Now, this isn't pertaining to COVID-19, but it is a coronavirus. So what I'm referring to, the common cold doesn't necessarily have a cure, which is also a coronavirus. Yet they have been able to rush something out, which is 90 to 94%, 95% effective for something that has a survivability rate of 99%. Okay. And they're going to rush this out. And of course, a lot of people politicians, government officials are expecting you to take this. Now, to sit up here and try to claim that someone's anti-vax because they're like, there ain't no way I'm putting that in me. At this point, I just don't care. And I'm not going to sit here and get in a dispute with someone calling me anti-vax, because my position is of great skepticism. Not even skepticism. It's a hell no. That's just what it is. Just a hell no. It's a hell no. Like, y'all should be free to take it if that's something that you want to do. But as it comes to me, I ain't taking it. My body, my choice. But let's check this out. Vaccination cards. This isn't Orwellian at all. Will be issued to everyone getting the COVID-19 vaccine, health officials say. The Department of Defense released the first images of a COVID-19 vaccination record card and vaccination kits Wednesday. This is reported by CNN, Fake News Network Health, says vaccination cards will be issued as the simplest way to keep track of COVID-19 shots, as Dr. Kelly Moore, Associate Director of the Immunization Action Coalition, which is supporting frontline workers who will administer COVID-19 vaccinations. A Department of Health and Human Services employee holds a COVID-19 vaccine record. This is what you're viewing right here. Because everyone will be issued a written card that they can put in their wallet that will tell them what they had and when their next dose is due. Let's do the simple, easy thing first. Everyone's going to get that. Vaccination clinics will also be reporting their state immunization registries, what vaccine was given, so that, for example, an entity could run a query if it didn't know where a patient got a first dose. This is weird, okay? As I'm reading this, like I'm not reading like a video game script or something like that, like the introduction to the zombie apocalypse or something on Resident Evil Umbrella Corps. That's not what I'm reading right now. This is being reported directly from Fake News Network, talking about, again, the Department of Defense and this whole Immunization Action Coalition. So as I'm reading that, please take into consideration that this is real life. Moore said that many places are planning to ask patients to voluntarily provide a cell phone number so they can get a text message telling them when and where their next dose is scheduled to be administered. Every dose administered will be reported to the U.S. Centers for Disease Control, the CDC, and Prevention. And Claire Hannon, executive director, said Claire Hannon, executive director of the Association of Immunization Managers. The CDC did not immediately respond to CNN's inquiry about whether such a database would include a record for everyone immunized. As far as vaccine kits, the DoD image shows the kits include a card, a needle, and a syringe, alcohol wipes, and a mask. Operation Warp Speed has 100 million vaccine kits, which is, again, you say what you will about Trump, he's complicit in all of that, because they've been discussing this, and he's talked about military folk being ready to go to make sure we get this out to everybody to get folks needles put in them. Was distributed a coronavirus vaccine starts, as Warp Speed Chief Operating Officer. The images come from two companies, Pfizer and Moderna. A panel with U.S. Food and Drug Administration is expected to discuss whether to authorize EUAs December 10th and 17th. Should Pfizer and Moderna get the authorization of vaccines, first shipments could happen 15 and 22nd, respectively, according to a document of the federal government's Operation Warp Speed. Initial supplies of the United States will be limited, and the wider population won't have access to vaccines until months into 2021. It doesn't matter. It's fine, because I'm not taking the shit anyway, so take as much time as you like. By February, 100 million Americans could be vaccinated against the Modelo virus. I will not be one of them. Now, look, we're talking about vaccine cards that are going to have to come directly from them. We know that. Okay, gotcha. But the problem here is that for those of us who aren't stupid, I guess, we know that that's going to open the door wide open for a papers, please sort of situation. People have put their entire faith in these vaccines, usually because they're sheep. And they believe that this is the end all. So whether it be from the government, we just talked about Joe Biden and the mask thing. But so whether it be coming from the mayors or the county judges or the governors or what have you, you know that there's going to be a situation where, again, as we talked about last show, there's going to be applied pressure on people who operate places of businesses, profit, nonprofit, doesn't matter. Places where people gather. And what they're going to do is place pressure on them. It's going to come from the government by way of that. And then it's going to manifest in ways that's going to make it very difficult to navigate the world, navigate your state, navigate your city, your county, in the event that you aren't vaccinated. Now, I sit and think, like, what the hell does it matter if you're vaccinated? If the vaccine works like you claim it would, you shouldn't even be worried about me or the people that are going to say, yeah, I'll take my chances. 99% survivability rate, above that with my age group. I think I like my chances here. Shouldn't even matter. But for whatever reason, they are pushing this idea that you must get injected with this and it's gonna manifest itself in ways, can't talk today, that are going to make it difficult to navigate, which is going to incentivize people to get it. I don't foresee them having an instance where you are directly forced from them, so government to individuals, saying you must have it. It'll manifest a way and effectively be that, but it'll be, again, them applying pressure to businesses and what have you, and then that manifests itself, and then it's like, hey, papers, please, and if you don't have your papers, you won't be able to do this, this, and that. Like I said, it sounds very freaky reading some of this stuff, that, you know, this is something that a real process sounds almost like, again, a video game setup, right? Is what it sounds like. I'm not taking a vaccine, and if I go to jail, that's what they're gonna have to do, essentially, for me, is force me to you know, they're gonna have to like hold me down and put that like stuff, like inject me with the needle. That's what's gonna have to happen. It's just something that I do think is worth going to jail over in the event that that happens, or and getting into beefs with law enforcement in the event that that's what it comes down to. But for people to so loosely and easily just say, yeah, we got to do it and we got to force it, really goes to show how people don't value actual bodily autonomy at all. Even coming from the individuals who would claim that they do on any other given day when it comes to like abortion or something like that, it's my body, my choice. When it comes to anything else, though, that logic doesn't apply. Then they think that the state owns your body. This is why that whole phrase was always bullcrap when it came from that particular side. Nonetheless, the more we talk about this, the freakier that it gets. And if you're one of the 100 million Americans to be vaccinated, congratulations. I hope the best out of all of this. Seriously, I hope that you don't end up with an instance that you grow a baby leg out of your neck as a side effect for the vaccine or something like that, that they couldn't foresee, because the baby leg don't start growing until 2 to 3 years down the line. Okay? Now, if that's what happens to you, that would suck. And I would hope that doesn't happen to you. And I'm not saying that it is. I'm not taking my chances. I'm going to watch y'all let that stuff play out. And when it comes to absolute freedom, that should be the case. Whereas if you want to take this vaccine, you should be free to take it. If you don't want to take the vaccine, you should be free to take it. And it shouldn't even matter, considering the fact that if the vaccine is effective, you're good if you're taking it. So what you worried about me for?
It's just incredible, actually. Obviously, I can't hold them in my hands because they are minus 70 degrees, but to know that they are here and we are amongst the first in the country to actually receive the vaccine, and therefore the first in the world, is just amazing. I'm so proud. They're stored in the minus 70 freezer, and they will be taken out of there when it's appropriate. The hospital is just going through the final checks to be able to implement the vaccination program for our high priority groups, and we will roll out when it's appropriate.
January 553, expiry February 21.